Hi, my name is Eric Yttri at Carnegie Mellon University, and I'm happy to talk to you today about a pair of algorithms now combined into a single platform, A-SOiD and B-SOiD, the two sides of a behavioral extraction tool that is useful for extracting behavior from all sorts of organisms as well as in all sorts of different contexts. I'd like to start off by thanking the members of the lab that contributed greatly to these efforts. These are Nahom, Jens, who is in Germany at Bonn University, and in particular, Alex Su, who recently graduated from the lab. And so one of our big questions, and arguably one of the bigger questions in neuroscience, is how to study the neural correlates of behavior, or how to study behavior in general. And so a considerable amount of work has been done using artificial and overtrained tasks. Here we have a mouse who's head fixed from our own lab, who's using a joystick, and the animal has learned that it will receive a reward for moving the joystick past a certain direction. And this is all fine and possesses some great experimental potential, but the artificiality of the task, the highly repetitive nature, and other issues may make this suboptimal. Another more abstract issue is that trials, as we like to think of them, don't necessarily exist, and that this reduced behavior that we're studying may produce a loss of dimensionality and also the neural encoding that we wish to study, or just artifacts from being overtrained. And so the alternative would be to study more naturalistic or spontaneous behavior, looking at an animal doing whatever it is that they want to do given a certain scenario. However, up until recently, this has been very difficult to do, particularly in any sort of high throughput fashion, due to the richness of naturalistic behaviors. Here we have Tala and just a subset of all the behaviors a dog can perform. And so we sought out to create an open source user-friendly platform in order to try and extract these behavioral syllabi from video. And so one of the major advances in the field as of late is computer vision algorithms such as SLEEP and Deep Lab Cut, as well as Deep Cut and others. But this information, where, say, a paw is only minimally interpretable. An animal may be performing any number of behaviors while having their paw in a given position. And so we sought out to add interpretability and to really pull out what behaviors are existing. And we see here with just 6 points, we can by eye tell when a mouse is walking, looking left, sniffing around, or grooming. And so a few years ago, we came out with B-SOiD, which is an unsupervised machine learning algorithm that is designed to learn and detect spatial temporal patterns in terms of movement. And the idea is that when walking, your paws and body are going to have one set of spatial temporal patterns. But when grooming or doing any of these other behaviors, the patterns will be different enough. And importantly, this algorithm uses the output from pose estimation. It can really use any type of pose estimation data. And it discovers, you don't have to tell it anything, it discovers the conserved spatial temporal patterns, and the categories that you see on the bottom of the screen are assigned by the experimenter. The algorithm doesn't know what walking is, doesn't even know what a mouse or a monkey or a human are. And so we've packaged this into a user-friendly, easy-to-download package that takes you step by step. And this slider here is really the only thing that an experimenter needs to manipulate. And the slider indicates if you want to be more of a grouper or a splitter when it comes to your behavior. You see here, it very rapidly will go through the data and cluster into these independent groups. And there's subsequent steps that allow you to get a full readout of the kinematics of those behaviors, as well as several sample videos of each one of those different clusters. And depending on this, you can go back and update the algorithm as necessary. So now I'd like to briefly take a look at how the B-SOiD algorithm works. So the idea is, again, that we can take in the spatial temporal patterns from pose estimation data. This is the distance between points, the speed, and the relative angle. One recommendation to potential users is that the information be independent. For some reasons, people will put multiple markers all along, say, one forearm bone. All those points are going to be moving essentially together. And so this redundancy in points doesn't do us any favors. Similarly, we recommend only labeling points that will likely be informative for your behavior. Because of this, we recommend that most users don't label anything except the base of the tail. The base of the tail in this image shown as the darker orangish red dot, because the end of the tail may move back and forth without any real relationship to the behavior itself. B-SOiD then takes these behaviors or these spatial temporal features and clusters them, identifies those conserved patterns of distance, speed, and angle. It can do this in 2 dimensions or in 3 dimensions. And then importantly, what B-SOiD does differently than previous attempts at behavioral segmentation is that it applies a random forest learning system in order to learn what the defining features of each one of these behaviors are. So for instance, you can see that on a face groom, the inter-forepaw distance, the distance between our forepaws, is going to be much less than if you are turning left or rearing up. These are histograms for each of those behaviors. And because of the random forest, we're able to generalize across different animals. We're able to apply this very quickly with less than 5 milliseconds of delay added by this algorithm. And also, if a particular specific combination of parameters hasn't been observed by the algorithm before, because of the classifier that we're using, it's not a problem. It will take what information it has and make a best decision. Also important, any 2D camera can be used, and because of how it's integrating information, we can actually derive 3-dimensional behaviors like rearing, and we can deliver the quantification, the classification of behaviors at the same temporal resolution of the camera used. So if you're using a 60 frames per second camera, you'll get behavioral output at a resolution of 60 frames per second. So here's some examples of the algorithm in use. For one, we took animals that had their A2A neurons in the striatum, this is the indirect pathway neurons, lesioned, and we just looked at their behavior. And because of the way B-SOiD is applied, we were able to identify different types of head groom. Again, B-SOiD identified these as independent behaviors, even though head groom and paw or face groom have been well-known canonically different behaviors. It was able to detect these. And if we look at just this head groom, which is a larger motion, we saw no difference between lesioned animals and control. Lesion in red, control in blue. However, if we looked at these smaller face groom type behaviors, we saw that the stroke distance, so every back and forth of the groom, the stroke speed, were both significantly shifted, as well as the bout duration itself. For those of you who study grooming behaviors, this is a level of detail that is very difficult to observe, both the separation of the canonical types of grooming, as well as the exact kinematics. Typically, labs will just study the duration of the grooming bouts themselves. So without this algorithm, one might assume that there is no effect on of the lesion, but in fact, there's a very, very strong effect that's particularly affecting the smaller movement type, smaller action type, and even within that, the smaller movement distances and speeds of that type. Another example, data here was applied to an animal learning a reaching task. So not necessarily a totally naturalistic behavior, but we see that sub-actions that comprise this reach to grasp task in data given to us from Dan Leventhal's lab at University of Michigan, we're able to show that through successive training sessions, that a pattern of organization grew out of the sub-actions that comprise a simple movement of reaching and grasping, and that the transition shown as the length, the transition probability shown as the length and darkness of the line increased over time, so that we went from fairly random assortment of sub-actions into a very stereotyped progression as training went on. Applied to humans, B-SOiD was able to learn sign language, in this case, simple finger spelling. Again, without telling it what any languages or any symbols meant. The idea here was just to test how robust B-SOiD would be to detecting these spatial temporal motifs. And finally, applied to the organism in general, here we have Andy, recent graduate from the lab. This is video just taken with a cell phone camera, which itself can move around, and using CMU's open pose. We can see that Andy, whether he runs with high knees or low knees, all those actions are assigned to group 0, whereas when he transitions to jumping jacks, we actually get two different actions, the upward phase and the downward phase of the jumping jack. This is because B-SOiD is detecting these as two different types of behaviors because of their kinematic differences. Finally, for B-SOiD, we have developed and are about to release a real-time version of the algorithm, which will allow experimenters to detect the behavior of choice and either induce a triggered object-genetic response or the stimulation otherwise, whether it be the presence of a food pellet or some sort of aversive stimulus. So all these behaviors are now accessible at lag times of only a few tens of milliseconds from the photon hitting the camera to the trigger being delivered. So that's B-SOiD, the unsupervised algorithm. I'd like to also introduce our supervised approach, which was just published in Nature Methods. And the reason for this is that unsupervised isn't a one-size-fits-all learning approach. The benefits are that it has minimal user input, so the effort is quite reduced. It also eliminates the possibility of user bias, but it only discovers obvious patterns. And so for individuals who are studying social behaviors or more nuanced or rare behaviors, having an expert definition of these behaviors of interest can be very useful. And in the case of social behavior, when we applied B-SOiD to multiple animals, what we found were just combinations of walking and rearing, walking and grooming, rather than the interactive type behaviors, such as investigating and attacking, that are more often studied in social dynamics. And so briefly, how A-SOiD works is it applies an active learning mechanism to deliver the behaviors of interest with up to a magnitude of less training data. And this is really the downside of supervised approaches, is the amount of training data that it takes to teach the algorithm the behaviors of interest. And so here in the schematic, a user would start with a very small amount of data. We recommend about 1% of the data itself. And then iteratively, A-SoiD will come back to the user and identify only the specific behaviors that were of low confidence. And the idea here is that you learn more from your mistakes, from your successes. This is also how we get to greatly reduce the amount of training data, because it is specifically asking for training on the difficult, the uncertain examples. The learning rate of the algorithm is greatly improved through these active methods. And so this delivers us a targeted, through this targeted iterative refinement, we quickly get accurate classification. And in the current version of A-SoiD, we actually combine the power of unsupervised discovery. And so you can take one class, say this blue behavior, and apply B-SoiD to this identified behavior, and we now have the ability to identify sub-behaviors, clusters within that, that gives you an even greater detail in your behavioral classification. And I'll have an example of that in just a couple of slides. In terms of our performance, we used the COMS-21 challenge data set from Ann Kennedy and colleagues. We found that compared to using the full 14,000 frames that were available to us, that A-SoiD was able to outperform the performance you would expect out of the full data set after only around 800 samples. So that's only 3 iterations of the algorithm, and it performs very powerfully. Compared to other methods, we compared to some excellent methods called Simba, as well as deep ethogram, shown here in blue and purple, as well as to the top finisher in the COMS-21 challenge. And A-SoiD outcompeted all of these. And so here we have an ethogram on the left comparing the performance with the target, the annotated behavior being shown in black, and then deeper quantification of this on the right-hand side, with the top dashed line representing the top performer in the COMS-21 challenge. Getting back to our iterative approach of using A-SoiD and B-SoiD together, we took one of the larger behavioral classifications from this training data set called Investigate, applied B-SoiD, and we found very well conserved clusters, which when we plotted out into either just modeling after 2 different animals or taking the motion energy, so how one animal moved relative to the other, we discovered, without actually even knowing so well that these were behaviors, or genital approach, which you might be familiar with if you've got a dog, as well as mutual investigation. So these were subclasses 2 and 5 of Investigate. It's really showcasing the potential power of this method, not just to get you accurate and rapid clustering of the behaviors of interest, but also a deeper understanding in case, say, one behavior may be different between 2 different disease states or times of day, males and females. The integration of the unsupervised B-SoiD within A-SoiD serves as a powerful additional tool that we've included. These are only a handful of behaviors. We decided to look more broadly into 3D pose estimation in a non-human primate, given to us by Jan Zimmerman, as well as a large human data set consisting of over 60 behaviors. Here we have the non-social behaviors shown on the right here. And we see that A-SOiD is able to very quickly and very accurately deliver automated segmentation in held out data, with the monkey behavior on the left and the human behavior on the right. And just as with B-SOiD, A-SOiD, which you can download for free, and if you want to read more, again, it's on Nature Methods. It comes with a user-friendly GUI that is targeted to helping you use the algorithm. It takes you step by step, and also shown here in E are some baseline statistics of the behavior. So it will tell you the duration, the speed for every stride or every bout, if one is detected, and other kinematic variables. So in the last little bit of time, I want to show a full application of this platform to neurobehavioral data. So our lab is interested in understanding how motor cortex and striatum work together to create behavior, in this case, spontaneous naturalistic behavior. And so we inserted a neuropixels electrode into the caudal forelimb area of a mouse. So this electrode spans all layers of motor cortex, dorsal striatum and ventral striatum, allowing simultaneous recording of over 300 neurons. And we recorded both the neural data and the video data from multiple cameras, essentially 24/7 over the course of 2 weeks per animal. We didn't quite realize that this would be over 3 terabytes of data a day, and because it's continuous, we'd have 6 terabytes tomorrow and 9 terabytes the day after that. But we were able to overcome these data management issues to get a really interesting data set. So here's just a sense of what we have with the neural activity. This is from motor cortex, as well as the concurrent behavior on the right from the side as well as below. And we were able to ask the first question of just, does the brain even care about these behaviors? It made sense to us as human observers. Mathematically, they're rigorously defined independent clusters, but the brain and certainly motor cortex and striatum don't necessarily need to represent these behaviors in its own way. It turns out that the cortex and striatum robustly modulate their activity with respect to these behaviors. Here we see each row is the activity of a neuron, and we've split it between superficial and deep layers of motor cortex, dorsal striatum and ventral striatum, and rank ordered the activity about the time of movement. That's the central black line, and shown the activity which ranges from -4 to +4 standard deviations, or z-scores, both 1 second before the onset to 1 second after, which means that each row is, although each row is one neuron, it needn't be the same neuron between these different behaviors that are listed at the top. These are 13 of about 20 behaviors that we were able to identify. This is a very rich data set, and is also almost somewhat surprising that we saw such a robust modulation of activity across these behaviors and different brain areas. We can also look at other elements as well in terms of their population dynamics. And we see, for instance, that in motor cortex, different types of grooming all have a similar shape and occupy a similar state space for these different grooming behaviors. However, in striatum, there's a sort of kinematic, vigor-related dispersal, where face groom 1 is the least vigorous movement, followed by face groom 3, on up to full-on head grooming, which is maybe the most vigorous of all these actions. And so there are certainly population dynamics as well as individual neuron dynamics that are at play here. and that these representations may differ between motor cortex and striatum. In the last little bit of time, I'd like to get into our prediction model, where we apply just a random force again, but now using the activity in any 100 millisecond bin, 100 millisecond period of time, to predict which of 16 behaviors was being performed at that point in time. And so here we have an ethogram at the top, where each color and position top to bottom indicates a different behavior over 30 seconds. And so we're going to essentially create a B-SOiD that can predict which behavior is being performed. Now, if we look at the actual neural data, we see that the layer 5-6 of motor cortex, as well as to some extent close behind it, dorsal striatum, perform quite well. And we found accuracy levels of 50 to 60% in terms of adjusted F1 score. This is many, many times greater than the roughly 1/16 prediction value we'd observe by chance. I'd also like to say that in this decoder, we're going at it about it in a very, very simple way. There's all sorts of behavioral dynamics in terms of transitions and durations of behavior that we could use to improve this as a B-SOiD, as well as other elements of the neural activity that we aren't taking into account. And that's because we're really interested in just what are the sort of lowest hanging fruit that we can observe when it comes to this neurobehavioral relationship without any bells and whistles. So we're excited to be moving forward on this. And we're excited to also collaborate with anyone with this neural data and or the behavioral algorithms themselves, because a tool is only as useful as how the field uses it. One last summary, just to show the time scale of performance of our decoders in a slightly different applied metric using AMI. We still see a ramp up of our decoding accuracy, and this is shared across several of these motor areas. All right, so in summary, A-SOiD and B-SOiD, now joined, provide a rapid, accurate, and automated behavioral segmentation of discovered, in the case of B-SOiD, or user-defined behaviors, in the case of A-SOiD. Any camera can be used to study any model organism, and certainly this is a pattern recognition machine, where there are some people using this for everything between proteomics to a seismologist in California. Let us know that he's a big fan of B-SOiD. And finally, this platform outperforms the current state of the art, including MoSeq and other algorithms like it on both the supervised and unsupervised side, while also being incredibly user-friendly and open source. So with that, I'd like to thank everyone for their attention. Please do reach out to me. Email address is there, and we have a step-by-step tutorial at the website listed at the bottom of the page. Thank you.